Yes.
Oh,
Okay. So good afternoon, everyone. I will be your chair. I'm Michel Orit from Leiden University in the Netherlands, and I will be your chair for the first session. Before introducing the speaker, let me briefly remind you of the rules. So we are not allowed to ask questions during the talk. Unfortunately, that's too difficult. So wait, please, with your questions for the Q&A session at the end, which we'll try to keep 10 minutes at least. What else? So remain muted, please, during the talks. And to ask questions, then you can either unmute yourself and ask, or write something in the chat, and we'll ask you to ask your question. So this is, I don't know which issue of the webinar, but today we have two speakers, Vahid Sandoghdar first, and the second one will be introduced by the chair of the second session, that's Subhasis Adhikari. It's a special pleasure for me to introduce Professor Sandoghdar, Vahid Sandoghdar. He's at the Max Planck Institute in Erlangen. Vahid, we know each other for more than almost 28 years. So this seems incredible, but it's what it is. Vahid, well, is Iranian, but he studied in the US, first at UC Davis, and then he moved to Yale. There he worked with Ed Heinz and Serge Roche. So I noticed that Serge Roche was also your PhD supervisor. I didn't know that. At Yale, where he worked on basically on Casimir forces in atomic physics. So Vahid has a very strong fundamental physics background component. After that, he went as a postdoc to the École Normale Supérieure in Paris, where he worked in the group of Serge Roche. He particularly worked on the whispering Gary modes and the way to enhance light-matter interaction with these cavities. And then he moved to the University of Constance in the group of Jürgen Linek, where he set up his own group and actually developed near-field optics. So he had very nice papers from that time. In 2001, sorry, he became a full professor at ETH Zurich, and he beat another group there, and has many papers from very nice papers from that time. And eventually he moved to the MPI, Max Planck Institute in Erlangen, Institute for Light Science, in 2011, and is still there. Vahid is very much interested in fundamental physics. I said quantum optics with single molecules at low temperature, but in the more recent years, he worked a lot in the direction of, let's say, room temperature applications, especially biomolecules, biophysics. And recently he has set up a whole new endeavor together with the hospital in Erlangen, to apply the science of light to medical problems, medical and biological. So I'm very looking forward to what he will tell us about today, and I give him the screen. So please, Vahid, go ahead.
Michel, thank you very much. Also to Frank and other organizers for organizing this series and for giving me an opportunity to be part of it. Thanks very much for the very kind introduction. Indeed, Michel was my gateway to single molecule work when I was a postdoc in Paris. That's when I first heard about single molecule works, and I spent a week in his lab goofing around and trying to get the basics of it, and that's how that completely marked my career afterwards, because when I became independent, I started to set up experiments that go in these directions. And so today I'm not going to tell you anything about those works, because they were low temperature single molecule spectroscopy works that we still continue to do. But as Michel mentioned, we've become interested in more biophysical applications. And it turns out that the way we do these biophysical applications is quite different from the way we used to do our other single molecule work. Although there are some very close links, as you will see, hopefully. So since this is a photothermal webinar series, I'm going to talk about the work that would be more related to that, and that would be essentially on our work in the direction of interferometric scattering microscopy, iSCAT. Good. So let me now see whether I can activate my here we go. Let me get a laser pointer. Right. So before I go on, I'd like to thank and acknowledge people who are working in the group right now. The top row are permanent staff, and then the blue ones are postdocs, and the red ones are PhD students. Almost everyone is working on iSCAT related work, but iSCAT started a long time ago, and there are a number of amazing people who've contributed to this, and meanwhile are continuing amazing work also in their own independent groups. So again, the red ones are the ones who were PhD students at the time, and the blue ones are the ones who used to be postdocs in the group. So let me jump right in to the discussion that I want to have with you today, and it's all about sensitivity and detection of matter. So we want to know how sensitively we can detect matter, and at that point we have to talk about what kind of material types we have. So things that absorb and fluoresce, that's the story that started with single molecule microscopy and spectroscopy. But not every piece of matter absorbs and fluoresces. There's the kind that absorbs but doesn't fluoresce. And then there's a kind that doesn't absorb either, at least not considerably. And of course, it doesn't fluoresce either. But you have to remember that everything scatters light. And everything scatters light because everything has some electrons at some point, and the moment you apply an oscillating electric field, as you have in light, there is some kind of a response. And so the story is, or the question is, how can you detect then this amount of, or these signatures of these matters through the interaction that they have with light? So again, in the case of standard fluorescence microscopy that goes back to early 90s, it was a huge challenge to get this to work. But nowadays we look at it and we kind of say, well, it's quite straightforward. You come with light, for example, green light, and then you detect red light, and it's a piece of cake to filter the green light and only detect red light, so you only see the molecules that are lighting up. And that way you have a very good signal to noise ratio, and you detect single molecules. So, and this has led to a revolution in fluorescence microscopy that led to Nobel Prize. And it all goes back to this idea that you can have very sensitive detection of fluorescence down to single molecules. And if you think about this revolution, then you see that it was really revolution for a factor of almost 10 in terms of revolution, because standard kind of diffraction limited or nearly diffraction limited type of resolution was about 100 nanometers, and with super resolution type of things, about 10 years ago, you could reach to 20, 30 nanometers, but there's no limit theoretically how far you can go. But then you also see that if, as an optical scientist, your pride is to always do better, you have another 2 orders of magnitude to bridge if your aim is to do detection of single atoms using light. And indeed, this is theoretically possible, and we and some other groups are working on that. For example, if you do cryogenic super resolution microscopy, you can get down to a few angstrom in resolution. But this is based on fluorescence, and this is what I want to right now officially this by saying that fluorescence has problems. It's wonderful. It's come a very long way, but you can't rely on fluorescence forever. You have to do labeling. There's photo bleaching, and of course there's saturation, and therefore a single fluorescent molecule can only give so many photons per unit time. So your signal is actually on the low side. So there's reason to want to look for alternatives. So let's go back and see what happens if you put something like a nanoparticle or a single molecule in a light beam. Well, the light is impinged on this nano object, and this nano object in general could be absorbing light and therefore dissipating it, or it could be also scattering. Something that would be more of an instantaneous just re-emission of the light that comes in. Either way, if you were to look in transmission, you would expect less light because some light is taken out of the original beam. And that could be summarized in something that you would know as a cross-section sigma. And if you look deeper in your textbook, then you would see that the cross-section of a two-level atom or an oscillating dipole is simply given by 3 lambda squared divided by 2 pi, which is roughly lambda squared divided by 2. And now if you think about it, you see that this cross-section is actually very large, because the thing that you want to compare the cross-section with is the size of the beam that you have, and you know that you can focus light to the limit of something of the order of lambda divided by 2. So this fraction lambda divided by A should become comparable to 1 according to this picture. Indeed, you can get to that point, and there are some subtleties or little issues, but in general, that statement is true, and you should be able to get a very strong extinction effect from a single molecule. Now, this is the only thing that I show you from the low temperature work. In the low temperature work, we actually can reach that limit of a very large cross-section, and that means that if you really do this simple experiment, that you focus light onto a single molecule, you look in transmission, you see that as you become resonant with that molecule, you can get a huge dip of 10-20%, because you have one molecule as a beam block. So this is to say that you don't necessarily need fluorescence to detect a single molecule. You should be able to detect it in a simple kind of transmission or extinction experiment. This is what we did also at room temperature. When you go to room temperature, you have to look in your textbook even a little more deeply, and then you see that ideal cross-section that you know for a two-level atom is actually augmented by this fraction gamma 0 by gamma, where gamma 0 would be the spontaneous emission rate of that two-level atom, but gamma would be the line width of that transition. And this line width, in the ideal case, would be gamma 0, so that fraction would be 1. But in real, this gamma is broadened because of all kinds of interactions that you might have. It might be quenching, but it might also be dephasing because of interactions with phonons. Either way, this ratio at room temperature turns out to be something of the order of 10 to minus 5 to 10 minus 6. And that means that this cross-section at room temperature is actually really small. But we showed in this paper that was published in 2010 that you can nevertheless detect a single molecule by doing a simple, what I would call a simple extinction experiment. So we had a Heine, you couple that into a microscope objective, you have a sample onto which you have spin-coated single molecules. And then you have another objective to collect all this light, and then you send that to a detector. And if you look at that signal now, I'm admittedly cutting corners on experimental details, but I want to just get the general concept across. You see that, again, you have a clear dip. This dip is only of the order of a few times 10 to minus 6 of the original intensity. But the important thing is that we're detecting this with a respectable signal to noise. And this is a signal from a single molecule, and if you waited for that molecule to bleach, then you would see that the signal indeed goes away. So all the details and control experiments are reported in this paper, but for you as a kind of a take-home message, I think it's useful or important to know that even a single molecule at room temperature can be detected in an extinction experiment, because this cross-section is small, but not ridiculously small. All right, you could do this now also to do imaging first, and second to do something like spectroscopy. So you see here, for example, an image of a single molecule that happens to be sitting here. If you were to illuminate it on its resonance, on its absorption resonance at 633, and this is what you would get if you were to go off resonance at about 670. And you clearly see that, again, there's evidence that all these other features stayed, but this feature here is no longer there. And indeed, if you subtract these two, you get a very nice signal from that single molecule by itself. Okay, so the situation that I've shown you is this situation that I have a light source, I have a molecule, and I look in transmission and I see the shadow of the molecule. This shadow of the molecule turns out, and you can also follow this very simply, that the way you would write the shadow or describe the shadow is that you have to write some kind of an intensity here. And the intensity would be the sum, the square of the electric field. Now, the electric field at the position of the shadow is the sum of the electric field that is incident and the electric field that's scattered by the molecule. So when you write it that way and now expand it, you see that you can write this as the intensity of the incident light, the full intensity, the intensity of the scattered light, full intensity. But then you have a cross term, which gives you the extinction term. And it's the phase of this cross term actually that leads to a destructive interference and gives you a shadow. Now, what you also want to remember now is that if you were to replace this molecule by a teapot or any macroscopic object, the physics doesn't really change. You still write the same equation, except that now you're not dealing with small amounts of light that is scattered by a molecule, but by a huge amount of light that is blocked by the teapot. So what I'm trying to emphasize is that the physics of normal bright field microscopy, which is exactly what happens here, and the physics of what I'm going to tell you about are actually quite common. So if we look at it a little differently, then you see that this is the experiment that I told you about. You come in, you focus your light onto a molecule, you collect that light and send it to a detector. You could do the same thing now by putting a camera on the other side and see the image of the molecule showing a dark spot. But what you can also do is you can put a slightly reflecting object here, for example, something like a cover glass, and reflect some of the incident light. And now you can show that on the detector, what you're detecting is the interference of the light that is reflected instead of the light that was transmitted, and the amount of light that is scattered by the molecule now in the backward direction instead of the forward direction. And again, you clearly see that the physics is the same, the equation is the same. You just have to worry about the quantities on this detector instead of this detector. And this is exactly what we realized when we were doing this experiment in early 2000s. And the reason we were doing this experiment is because we were excited about, or we were looking for ways to detect small gold particles as exemplary nanoparticles. But we wanted to show in the wake of all that single molecule work and fluorescence work, whether you could detect very small nanoparticles without using fluorescence. There was also some motivation to do plasmonics and all of that. But from a detection point of view, this is basically what we did. So this is the first work, and now if you want to read more about the details of iSCAT and how it evolved and how it connects to all kinds of other techniques, I really recommend this book chapter that came out a couple of years ago. All right, so what's the story with iSCAT, the way we kind of developed it? Well, in the early days, we had a gold nanoparticle and we wanted to detect it. Usually you would go and do dark field microscopy, but if you've tried dark field microscopy, you know that when your particle becomes smaller than something of the order of 30 nanometers, your signal goes down very quickly. But if we get down to the basics of the physics, then you see that we basically have something like a round object, but its size is much smaller than a wavelength. So you can describe this as a Rayleigh particle. And the amount of light that is scattered, the electric field, is given by the incident field times the polarizability. And the polarizability is proportional to the volume, that is the third power of the diameter, and a fraction here, a quotient, that has to do with the dielectric function of the particle and the dielectric function of the surrounding. All right. Now, we put this particle, again, at an interface. Usually, you don't have to do it, but this is how it's done in most applications. And when you illuminate that, you get a bit of a reflection and some scattered light. And it's these two fields that interfere with each other. And again, you have the same equation as before. And now if you look at it, you see that the amount of light that's scattered, the intensity of that, is proportional to E squared and therefore to the sixth power of the particle size. And this is the quantity that you would detect in dark field. But you also see why dark field loses its signal very quickly, because it scales as the sixth power of the size of the particle. So if you change the particle by a factor of 10, you lose a factor of 1,000,000 in signal. The extinction signal, this cross term, also, of course, drops as the particle becomes smaller, but it drops more softly. So if you change the factor by a factor of 10, you only lose a factor of 1,000, let's say. All right, so there's more to the subtlety of dark field versus iSCAT, but again, if you read that book chapter, you get the full description. I'm not going to go through that discussion right now. So what we do is we don't do dark field, which means we take the whole thing on our detector and rely on the fact that this contribution is going to win over this contribution for very small particles. That is, this part disappears more quickly. So these are some examples of the early iSCAT signals. We were very happy to detect small particles as small as something like 5 nanometers. But of course, these are 5 nanometer gold particles. And we also already noticed that, as you get to very small particles, your image becomes a little speckly. So I will come to that issue, but to summarize kind of what now happens if you compare iSCAT with fluorescence, you would see that, well, in iSCAT, you can get to a shock noise limited detection almost. And that would mean that the more light you have, the better your signal to noise ratio. And the fact that an object like a gold particle, or later we'll talk about proteins or other dielectric objects, do not bleach, means that you can put a lot of light onto them and therefore improve your signal to noise ratio in principle, indefinitely. And if you were to have a better signal to noise ratio, you would have a better localization precision in your imaging. And if you have a better signal to noise ratio, or you have more light, it also means that you can detect faster, you can integrate faster because you just have more photons. At this point, it's important to, I've mentioned this already, but the equation that I wrote down for iSCAT is really just an interferometric formula. And that's why iSCAT is just the youngest, maybe, sibling of all these other methods that have been around forever. and they all rely on some kind of an interferometry. The thing that we think has made iSCAT more special, or do things that these other methods hadn't achieved, although they've been around for several decades, is that we have put the emphasis on squeezing more signal-to-noise ratio and to squeeze the signal to the point of detecting nanoparticles instead of detecting, for example, a whole cell or larger objects as you would do in digital polography or quantitative phase imaging. All right, so the smallest that we've detected is a protein. So I'm gonna jump right onto that and then back off and talk about slightly larger particles. So in the case of a protein, this is important because you're interested in detecting proteins for all kinds of sensing applications and biomedical applications. A typical sensing device would have a surface, for example, that would be sensitive to the proteins that you add. Now, if we prepare a surface that is functionalized before and look at that surface without added proteins in iSCAT, then you would see this kind of a speckly signal. And it also shows you why it's difficult to detect to do iSCAT, because you, compared to dark field, you are not filtering anything out. You're getting all the information that there is from your sample. And of course, if your sample has slight index refraction heterogeneities or slight tiniest topography, you end up scattering from it and therefore end up with a speckled image as I'm showing you here. But then you realize that because this speckle has to do with your sample, it could very well be that your sample is just stable and constant, which means that if you subtract two consecutive images from each other, you can flatten your screen to the point that you can now detect incoming single proteins. And this is basically what happens now. That is, you let proteins come, and between frame 1 and frame 2, a protein has landed. And therefore, when you subtract those two frames, you mask all the background and only get the signature from the individual new proteins that have been added. So here's an example of a video that I run for you. On the left, you have the frames. Right now, the valve is closed. At some point, we open the valve, and then you would see that proteins gush through. And every time that they come, right they are. As they come, you see a dark shadow on the camera. And on the right, we just registered those dark shadows. So the advantage now is that you're not just doing protein sensing with single protein sensitivity. In addition to that, you actually know exactly which protein landed where and at what time. So this is what was published in 2014, and we're very happy about this because we've taken now the sensitivity of iSCAT, which is, again, the simplest form of imaging or the simplest form of detection that you could have, down to single protein sensitivity. And of course, in this form of sensing, you also have the great advantage that you don't have a small sensing surface, but your sensing surface is large because you don't have to look for something like a hotspot or a special region. Anywhere the protein lands, you can detect it. Now, we tested this for different proteins because you can look at larger proteins, for example, in this case, 340 kilodalton, or a smaller protein of 65 kilodalton. And as you see here, the smaller protein gives you a weaker shadow. And if you line up the contrast, then you see that they line up as they should along the line. Now you go back to the original the equation that I showed you for iSCAT. Remember that the signal that you have is proportional to the polarizability and therefore the size of the protein. And I'll come back to that in a second, but I also want to again emphasize that you have information about where the proteins land. And as before in localization microscopy, once you have a point spread function, you can find the center of that and therefore know where the proteins are landing And we could do that in this first paper with a precision of ±5 nanometers. Okay, so I showed you this equation, and we said that for very small particles, the direct contribution scattering intensity can be ignored. So the signal really just comes from this cross term, and now you have the background, but the background is in principle constant. So we've set up a contrast for iSCAT, and that would just be this whole thing minus the background, that is this part here, divided by the background. That's what we call a contrast. And if you do your little math, then you see that the ER cancels from top and bottom, and so you end up having this quotient here, plus this phase here, and the phase will play an important role in a bit. So the top part has to do with the polarizability. Polarizability has to do with volume, and if you are dealing with things that have the same density, for example, protein type of matter, you can assume that it's also proportional to mass. The part down here, how much reflected electric field you have, has to do with the substrate reflectivity, and of course there is some degree of freedom that you have there in terms of adjusting your contrast. And as I will show you in a bit, the phase here can be responsible for giving you information about the axial position, because as the particle goes up and down, there is a traveling phase that is accumulated, and therefore the contrast can get modulated from 0 or -1 to 1 if you want. Okay, so this idea of the polarizability being proportional to the mass, and the fact that the particle that the contrast lines up linearly, was exploited very nicely by the group of Philip Kukura and his mass spectrometry colleagues. And they showed and argued in this paper that you can really use iSCAT as a measurement method for quantifying masses of protein, kind of like doing mass spectrometry. But now you're doing it for objects that are larger than the usual very small molecules. And we've also shown that you can do this, for example, to look at the secretion of a single cell. If you have a cell, the cells quite often secrete proteins or exosomes. And so in this experiment, what we did was we parked our field of view next to the cell, and we wait for the cell to eject proteins, and as the proteins land within the field of view, we see their signal. So this is now a typical kind of iSCAT video that we record. The dark spots or shadows that you see, these are particles that are landing. And we do this experiment now over, let's say, 20-30 minutes, because the cell has activity that goes on for many minutes. So in real time, you can really watch this. And this is very exciting because it shows that you can now use this information and do all kinds of fancy experiments with your cell. You can activate it, you can manipulate it, perturb it to see what it does in real time. You can make histograms of the contrast that you detect over, again, whatever time you want. And in the cells that we looked at, we knew that they should be ejecting IgG, and through control experiments, we showed indeed that they are ejecting IgG. But we also see that the cell is ejecting other things, and this is the really exciting part. But quantifying exactly what this is something that went beyond that work, and right now we're doing experiments where we're trying to actually design experiments where we excuse me, try to use iSCAT to learn about secretion activities of cells. There's a lot of, you can call this something like dark matter. There's a lot that is secreted by proteins, but you don't know what that is. So this would be the first step in terms of documenting what the cell is doing. So now I go back a little bit in time to some of the work in the early 2010s, where we were excited about using iSCAT for particle tracking. Particle tracking, single molecule tracking, also goes back to the 90s or even late 80s, where biophysicists were interested, for example, in understanding the dynamics of diffusion on membranes. So what I'm showing you here is a model membrane, is a synthetic membrane, is a double layer lipid layer that you can fabricate on glass substrate. And we could label the lipids with small gold nanoparticles and now chase the lipids by chasing the gold particles. This would be a typical iSCAT PSF that we would see, and again, you can find the center of it by doing localization microscopy and therefore do tracking. What was exciting about this is that, again, you have to put it in context of normal single molecule fluorescence kind of tracking. You see here that we're resolving domains that are 20 nanometers large, but the individual localization events we're detecting with precision of the order of 2 nanometers. And that's why within something like 20 nanometers, which is a typical kind of localization precision in fluorescence microscopy, we can see hundreds of events and therefore figure out exactly what the object is doing in this small domain. This is something that kind of smells like a raft physics. In this case, there's no interesting raft because this is just a synthetic membrane on a glass substrate. Here's another example of even smaller domains that we could resolve. But to look at, let's say, unperturbed lipid bilayers, we spent a lot of time learning how to make these giant unilamellar vesicles, so-called GUVs, which are like soap bubbles. They could be just full of water inside and full of water outside, but in between you have a double lipid layer. And again, we can label that with a gold nanoparticle. And now we want to chase the gold nanoparticle on this curved surface. The typical diameter of a GUV would be, in our case, 20-30 microns. If you were to look at that GUV in fluorescence, you would see that it's indeed roundish. And if we put this GUV on our iSCAT microscope, we see, to start, you see Newton rings. You see Newton rings because, of course, iSCAT is an interferometric detection. And you already see interference. Now, not necessarily because something was scattered, but you can now describe this as reflection, as you would in your Newton ring physics. You have a curved surface, some light is reflected, and that light interferes with the light that is reflected directly from the substrate. And as you go up and out, the phase changes, and therefore you see Newton rings. But if you pay little attention, then you see that on this whole thing, there is a little spot here in front of my laser pointer. And that is the signature of a single gold nanoparticle. And when we run the movie and we, again, subtract the background that is, in this case, not 100% static because the GUV actually has a little bit of a wobble. But if you take care of that background, then you see the signature of the gold nanoparticle that is moving around. If you pay more attention, you also notice that the contrast is modulating from black to white. And that has to do with the fact that the GUV is now moving up and down. And as in the case of the Newton rings, the phase is being modulated. And remember that there was that cosine phi in the contrast, and that's what gives rise to this contrast. So now, by looking at videos like that, we have a lateral information by doing normal localization microscopy, but we also have Z information because we have information about the contrast of the particle. And by doing that, we can provide 3D trajectories, except that now we have nanometer precision in all directions. And this is quite exciting because you can now work on on a free membrane. Okay, so the last chapter I want
only
to
a
tell
few
you
minutes
about
to
quickly.
conclude.
Sorry?
Only a few minutes to conclude. The time is up.
This is the last bit that I want to tell you. If you take that GUV and make it into a real cell, then the background becomes more difficult, but the problem becomes more exciting. So this is the typical background that you would have because the cell is a crowded place. And but on top of that, you see that you still can kind of recognize a particle moving around. This particle has been bound to a transmembrane receptor. And by doing some image processing tricks, we can again get rid of the background and make trajectories. Let me not go through those imaging tricks, but you now see that we make trajectories. Again, you look at the scale, this whole thing is 100 nanometers. We have a very good localization precision, and that makes us, that gives us the possibility to track a protein over a long time of many, many minutes, but with a temporal resolution of a few tens of microseconds. And that means that we get a lot of trajectory points that if you were to compare with the typical fluorescence, you would clearly see that you have a lot more data. That data allows you to have quantitative statistics and indeed learn about real kind of confinements or raft type of physics. The details are in this paper. In this paper, we were demonstrating the methods, so we're not going into the physics of or biophysics of rafts, but we clearly see confinement events and subdiffusion events. Here's a story about how you can extract more information from the point spread function of iSCAT, which compared to fluorescence is a lot richer because you have all these interference rings. And if you look at the details of the interference rings, then you learn about the axial position beyond that simple modulating traveling phase. And this is what we've done to extract information, for example, to see how a particle moves really just by a few tens of nanometers on the surface of the cell while it's confined. You see clearly that it's not just going randomly somewhere. First of all, it has a certain sense of purpose in terms of angle, but also it stays on a kind of a cusp. And these are all the exciting things that we're now exploring, trying to actually use this now to learn some biophysics. And let me just go very quickly to the end to show you that the same thing can be done not just with gold nanoparticles as labeled, but they could be done with viruses. Viruses are very interesting objects that by now everyone appreciates, but we liked them already more than 10 years ago because a virus is a dielectric nanoparticle that gives you a quite large iSCAT signal. And of course, it is biophysically relevant. So we have now a strong line of work where we're trying to look at viruses, not just on synthetic membranes, but in real cells. If you're interested in iSCAT, I told you about the book chapter, but there's kind of a small version of that book chapter you can read in this mini review in iSCAT. And if you're interested in doing iSCAT, here's an open source package that we've put out, and we'll be augmenting this in the next few weeks. Where you can download and also manipulate as you want to do image analysis for the iSCAT videos and images that you record. And with that, I stop and be happy to answer any questions if there are some.
So thank you very much, Vahid. So I think I can upload from the audience. So I put my clapping, and I'm already asking for questions. So either speak up or write in the chat. And as I don't hear anything, I was wondering, maybe I can start with the first question. In iSCAT, can you look at other signals than just the presence of the molecule at the pixel in your image? But for example, the way this fluctuates. So have you tried to use this information to, for example, distinguish different molecules, or to have more information than just the mass of the molecule? For example, its shape or its rotational diffusion, etc. So have you tried that?
Right. Yeah, that's a very good point. So depending on your signal to noise ratio and your ambition, the answer is definitely yes. So as I mentioned, the signal in iSCAT has to do with the polarizability, but I presented that in the case of a round object. The moment you have an object that is anisotropic in space, the polarizability also becomes anisotropic. And that already tells you immediately that yes, if you had something like a nanorod, you would have a polarization response, and therefore you immediately become sensitive to rotational diffusion. Now, if you were to miniaturize a nanorod into something like a protein that is not just a fluffy ball, but it has a structure,
yep.
In principle, you can do the same thing. But of course, at that point, then you want to have a large enough signal to noise ratio to tell the difference between one polarization and the other one. And right now, I don't know of anything. There are some very interesting works by Marek Pliarek's group now, where they look at the anisotropy of microtubules. So a microtubule is considerably larger than a protein, but it's quite small, and the anisotropy of the microtubule is something that has been shown with iSCAT. So in principle, yes, but in terms of details, you have to look at your signal.
All right, thank you very much. Any more questions?
Yes, if I can have one question.
Yes, please go ahead. I don't know who you are, so please introduce yourself and ask your question.
Sure, that's my pleasure. I'm Sohail Mojiri from Professor Enderlein's group in Göttingen. Thank you so much for your nice talk. Actually, I've got one question regarding the laser coherency. To some extent, the laser should be coherent if you want to get really high contrast from your images.
Right. So considering that, so laser coherence, as you noted, or a certain degree of coherence is important. But depending on how you do the experiment, the too much coherence could also hurt you. So if your coherence length is very large, that means that everything in your sample over several millimeters would contribute to your speckle. So in principle, you want to keep your coherence length as small as possible, but small enough for you to be able to detect your signal. And now if your signal is in many of the cases that I've shown you, regarding a particle that is very close to your substrate, then of course you need almost no coherence. You can do it with incandescent light, because there is no distance between the two parts where the light gets reflected, so they're always coherent. But if you if your particle is, let's say, hovering around at 50 microns above your substrate, then of course you want to make sure that your coherence length is long enough to be able to do that type of interferometry.
Okay, yeah, thank you so much.
Any more questions?